Recording begins. It is October 8th, 2025, and this is Biology 105. Thank you all for making it to class again. It's so good to see you all here. We're in week 7, so if you don't count fall break and you don't count finals week, week 7's your halfway point. This is Wednesday, so it's like halfway through the semester, believe it or not. All right? It's pretty exciting. Now we just got to make it through the other half. All right? So this week, we're going to wrap up our week 6 material, start our week 7 material. You know the drill. Don't forget your reading quiz exists. Don't forget that you've got a lab assignment. All right? Very good this week. It's all about enzyme kinetics, which we'll finish talking about today during the lecture. So it's going to be a nice lab. It always works. I want to point a couple things out about the lab, okay? You've got this enzyme kinetics quiz where you have to graph some data. And as per usual, I include a tutorial video where I show you how to do the graphing in Excel. All right, so it's right there. There's the video even embedded in the assignment itself, okay? You're going to be uploading some graphs. All right, uploading some graphs, answering some questions, that's it. Pretty straightforward. Definitely watch the video as you go. I also have a data sheet where you can put in your potato data, okay, that you generate from your enzyme lab, and then you can graph it for fun. I have it all set up to do the conversions and then generate figures for you. It's a great time, all right? This is one of those things where doing this, graphing your own data, is going to be very helpful when you hit the lab midterm assignment and the lab final, and you have to interpret one of these figures, okay? So I'm strongly encouraging you to do this, but it's not necessarily part of your, sorry, this is from the osmosis lab. You already did this one. My bad. Disregard that statement. I don't know why I had that open. It's because they both have to do with potatoes. So I just got confused. Apologies. Enzyme kinetics. Follow the video. I give you the tutorial. You're using data that you collect in class as well as some data in your protocol. So you're going to hit a point where you're like, experiment 3, we didn't do experiment 3. But just look at the assignment and it tells you, and in the protocol it shows you, experiment 3's results are in a table. Okay? So the data's right there for you. I just want to make that announcement in class to avoid a bunch of panic. Okay, cool. Week 7, enzyme kinetics. Week 8, online lab midterm, worth 50 points. All right? You still have a regular lab and a regular lab assignment, but there's also this online lab midterm. All right? You have one attempt on it. Use your lab notebook. I know I talked to you about it on Monday, but don't forget this is coming up because it's a great time. It's not too late to start making your lab notebook. Heck, make it this weekend. You need something to do on the weekend, right? Yeah. Okay, cool. Any questions before we just get back into the lecture? We're actually right on track with where I was hoping to be, so we're not on a time crunch right now, which is rare. Yeah, it's fine. Oh yeah, look how far we are in this PowerPoint. My goodness. Okay, so if you remember, folks, I was talking to you about, hey, listen, enzymes are proteins and proteins denature, okay? So if you get an enzyme into an extreme pH or you get an enzyme into an extreme temperature, all right, it's going to stop functioning, all right? Enzymes have ideal temperatures for their operation, and they have ideal pHs for their operation. Fair enough? So different organisms, okay, their enzymes could have different ideal temperatures based on the environment they live in. And within a single organism, enzymes that are in different parts of your body that have different conditions, they might have different ideal pHs or ideal temperatures. Okay, but it's usually a pH thing because the temperature throughout your body doesn't, only varies to a limited degree. Any questions about this stuff? I talked to you about the interesting aspect of, molecules are moving around more, so it ramps up and then it denatures, so it just falls off. But that's not something you're going to be tested on or anything. It's just fun. So let's move into new material. Okay. hey, listen, if you put something into an extreme pH, it denatures. We're good? Okay, cool. What can affect enzyme activity? Well, we've already talked about temperature and pH. There's also the very general bullet point here of chemicals. All right? I know. So certain chemicals can react with proteins and make them sort of lose their shape. If a protein loses its shape, then the enzyme's not functioning anymore. Okay? Especially if the active site loses its shape. And now I'm trying to prime the pump here by talking about the active site. So remember, the active site is what actually binds the substrate, and it's what's really responsible for the activity of the enzyme, if you're willing to walk with me down this oversimplified path, which is BIOL 105. All right. But we've also got these biological factors. So things that are made in the organism to regulate enzyme activity, or things required by the enzyme to be active. Biological factors is kind of a weird category, but bear with me here as we talk about them, okay? Sometimes an active site, a protein's active site, it can't do its job unless there's some additional compound there, like a charged ion or something. Maybe like an iron ion, all right? Without this additional piece inside of the active site, the enzyme doesn't function, or it doesn't function very well. So these things are called cofactors. We're going to talk about them more as we move forward, but in heme, the iron atom is responsible for binding oxygen, all right? It's a whole chemical thing. Next, so far we've been talking about one thing that interacts with the active site. This is like a non-protein enzyme helper called a cofactor. It gets into the active site, helps it do its thing. We've also got these mechanisms for regulating enzyme activity, which all come down to put something else in the active site, and that means that the active site won't function, okay? Or change the shape of the active site so the active site doesn't function. So these are the inhibitors. And inhibitors work by binding to the enzyme and preventing the active site from working. They can do it in two ways. They can compete for the active site. Those are the competitive inhibitors. Or they can bind someplace else on the enzyme and cause a change in the enzyme's shape, changing the shape of the active site and preventing it from being able to carry out its function. Don't worry, we're going to talk about competitive and non-competitive inhibitors with examples, with visuals and everything, okay? So, great. Enzyme activity can be regulated. That is our key takeaway here. Cool? Keep on moving forward. Metabolism, all right? We've already sort of talked about this, but you know how much I love repetition. It's all about bringing it back. Oh, question, please.
So the cofactors help the enzyme.
Mm-hmm. Yeah.
Like a helping hand.
Yeah. It's not a protein. It's not part of the enzyme. We don't necessarily make it internally. We can't synthesize it in the same way. So it's oftentimes, it's like a vitamin that you take, just works as a cofactor in enzymes.
Helps the reaction.
Yeah, think about it like you've got the active site, but it needs like a supercharged ion in the center of it to actually carry out its function. If that, yeah, exactly. The active site's still there. It can still bind the substrate, but it doesn't have that supercharge right there that's going to really make it happen. Of course. Okay, metabolism, coming back. What's metabolism? It's all the chemical reactions that occur inside of a cell, inside of an organism. It all depends on how you're defining the metabolism. The metabolism of a cell is all the chemical reactions going on inside a cell. The metabolism of an organism is all the chemical reactions going on inside of the organism. All right? Metabolism. So we talk about these chemical reactions inside of cells or organisms in metabolic pathways. They're usually these multi-step pathways where a molecule starts here, and then an enzyme does something and it's changed form, and then a different enzyme comes and does something and it changes form again, and then a different enzyme comes and does something and it changes form again, and now it's in its final form. Sometimes these breakdowns are to release energy, sometimes these breakdowns are to produce one thing from a precursor molecule, sometimes these pathways branch in all sorts of different ways, where the single starting molecule can make a variety of different products depending on which enzymes act on it. So metabolism gets really complicated. It all is usually discussing multi-step metabolic pathways though, all right? Where there's multiple conversions, where it becomes substrate to product, and now it's the substrate for a different enzyme, which makes it a product, and now it's a substrate for a different enzyme, which makes another product. Cool so far, folks? So what I'm adding in here to our previous metabolism slide is enzymes, all right, catalyze each step in the metabolic pathway. I keep stepping on this vent and it squeaks, and I'm like really in my head that you in the front think I'm farting, and I'm not. It's this squeaky vent right here, I swear. Anyway, now I've got it out there and I can focus on the lecture. I just had to get it out of my head. Okay, cool. So he's definitely farting, right? Yeah. So we've got catabolic and anabolic pathways, which we've already talked about, but I haven't given you my fun mnemonic device, all right? So catabolic pathways, remember, are breakdowns. They break something down and they release energy, okay? Because you're going from more order, right, to less order. Sometimes it's hard to remember catabolic and anabolic. I think about it like this. If I build this up and there's a cat present, the cat will come by and knock it down, okay? So the catabolic pathway is the breakdown product, the breakdown pathway, because cats break things apart, you know? If that works for you, that's great. It's always worked for me as a memorization thing. Some people say the T looks like a plus sign, and they use that, but I don't like that as much because it's pluses and minuses. It's confusing. Anyways, catabolic pathways, anabolic pathways. Anabolic pathways take a bunch of little things, build up something big. This is why anabolic steroids are called anabolic steroids. They build something up. Okay? Questions? Let's continue moving forward. Here's a multi-step, generalized metabolic pathway. Check it out. Starting molecule, enzyme 1 makes molecule B, enzyme 2 takes molecule B, makes molecule C, enzyme 3 takes molecule C, makes molecule D. Cool? Each one of these enzymes' activity can be regulated in a number of different ways. And oftentimes, these enzymatic catalyzed metabolic pathways are really, really complex, okay? So they branch all over the place. There's cycles where things shoot off. Like, it's just incredibly complicated and frustrating. But really, the whole thing is, there's like an enzyme doing this, and then look right here, there's a branching point, and it can become this and this, but we need this precursor to react with that to make this, or these two precursors can both become the piece that feeds in right over here, you know? It's fine. If you're doing biochemistry, you're probably going to focus on a very specific metabolic pathway and spend a lot of time learning it, but otherwise you can always reference a full chart like this. So it's very complex, but we're going to make it as simple as we possibly can. Sound good? Okay, I'm stoked. There are all these metabolic pathways that could be occurring, but they're not all occurring at once. All right? The cell's metabolism, the organism's metabolism needs to be tightly regulated. How do we do this? Well, one of the most simple ways is to regulate the genes that are being expressed. If the genes that encode the enzymes aren't being expressed, the metabolic pathway can't happen. So that can lead to differences in development. It's part of the differences between different, cell types and tissue types. It's all dependent on which enzymes are present in each cell type, because that determines which metabolic reactions are happening. Bless you. That determines which molecules are present, right? So certain cell types will have certain enzyme genes turned on or turned off. Does that sort of make sense to folks? Bless you. And then, once you've got the suite of enzymes that are normally there inside a cell, now you can further regulate the actual enzymatic activity by, say, using inhibitors. And we're going to talk a lot about that. So right here, we've got tryptophan and the multi-step metabolic pathway for generating tryptophan from a precursor molecule, which we're just going to call a precursor molecule. Okay? Totally fine. Check it out. 1, 2, 3, 4 different molecules. All right? Converting precursor to molecule 1 is carried out by enzyme 1. Enzyme 1 has quaternary structure, so technically there are 2 genes involved in making this enzyme. Remember, the quaternary structure is when multiple polypeptides join together. Okay? Enzyme 2 is a single gene. Enzyme 3 is 2 genes. Boom, boom, boom. All right? Now check this out. Bless you. Tryptophan is the end product. When you've got enough tryptophan, you don't want to keep making tryptophan, because this precursor molecule, it probably branches off and is used in a ton of other metabolic pathways. By the way, you have to memorize this and recreate it on your final exam. Very fun. Ha ha ha, it's just a joke. Yeah, very good. All right. So you don't want to waste your precursor molecule. Plus, these could be requiring energy. So check this out. This is really, really standard, and it's a process that you need to know about. This is called feedback inhibition, where the product of a metabolic pathway inhibits that metabolic pathway. So tryptophan itself right here, the end product right here, is somehow inhibiting the activity of this enzyme that's responsible for making tryptophan. The textbook example that I just don't like very much is the thermostat. And they'll be like, your thermostat on the wall, it makes heat until there's enough heat, and then the product of the thermostat, which made heat, inhibits the thermostat's activity and it stops making heat. I don't like that because that's not how a thermostat works, but you know, that's neither here nor there. Here though, in the metabolic pathway, tryptophan inhibits its own production. And this is great, this is all over the place. Here's the benefit, okay? 1, you don't make excess tryptophan. And 2, when you've used up all the tryptophan and you need more, that inhibition gets released. So it's sort of a self-regulating process that always maintains a certain amount of tryptophan. Reasonable? Question?
So I just want to make sure it inhibits the enzymes, not the precursors, right?
It inhibits the enzymes. The precursor here is the substrate for this enzyme. Yep, very good, very good. All righty. I talked about all this already. I forgot, honestly, I was so focused on the figure, I forgot there was actually text on here too. Yes, please.
Can you just repeat your definition of feedback inhibition again?
100%. Ready? Here it comes. In feedback inhibition, the product of a metabolic pathway inhibits the metabolic pathway. Yeah, no problem. Yes?
Can you go over the regulation of enzyme production just one more time?
What the hell is that? I'm sorry, what the hell is that noise? Oh, I did not mean to cut you off there.
A couple weeks ago, this one short-circuited, so it's...
Oh, okay, thanks. I thought somebody was like rubbing their fingernails on one of the chairs, and I was about to lose my mind. Yeah, like that, yeah. All right, so sorry, please.
So can you just go over the regulation of enzyme production again?
Yes, yeah, totally. So one of the ways that, say, like cell types are determined is what enzymes are even present in those cells, okay? So, man, that's really bad and annoying. Yeah, I can do this, we can do this as a group, all right? Let's dial in, here we go. Enzymes are proteins encoded by genes, all right? One way to determine if a metabolic pathway is going to happen is, are the enzymes even present, all right? If you don't make the enzymes for tryptophan production, then tryptophan doesn't get produced, right? So if you have certain cells that don't need to make tryptophan, then you just never express these enzymes inside of those cells. But in other cell types, you need tryptophan sometimes, and you need to regulate how much tryptophan you're making. So that's where feedback inhibition comes in, where when you have enough tryptophan, you stop making tryptophan. When you don't have enough tryptophan, you make more tryptophan. All right? Now, sometimes the tryptophan itself can regulate the genes that produce the enzymes for making the tryptophan, but that's a way more long-term process because those enzymes that were already made, they're going to stay there and keep operating and keep making tryptophan. So this is a long-term process. This is short-term, all right? And this pathway here, I mean, it's kind of like more, sometimes it's used more by like prokaryotes, bless you. I'm getting getting into way too much detail on this, honestly. But the gene expression piece, did that sort of make sense?
Kind of.
Yeah.
It's basically the same process, but you're using, and you're making enzymes instead of regulating the activity.
Exactly. You're making more enzyme or you're not making enzyme, as opposed to you're blocking the active site or preventing the enzyme from working. Yeah. Very good. I wonder if I can audio mute and that would do anything. Okay. Cool. So folks, I can't overstate how important enzymes are. It's easy to think of them as like, oh, this is just one aspect of the cell, but hey, listen, your cell isn't all protein. You're not all protein, okay? All of those other chemicals that are different between cell types, okay? Whether they are present or not, and in what proportion they are present, is all due to the enzymes that are present, okay? So the metabolic pathways that are happening determine the non-protein components of your cells. And those non-protein components, I'm sorry, I can hear you. And the non-protein component, okay, is all determined by what enzymes are present, all right? So enzymes are so dang important. Cool? I'm sorry, I can still hear you. I gotta get you to zip your lip. It's so distracting. It's so distracting. Okay. Good deal. Ready for feedback inhibition in a slightly different way. A, B, C, D. Here's the reaction, enzyme 1, 2, 3. All right? Enzyme D is going back and preventing, product D is going back and preventing enzyme 1 from acting, therefore inhibiting its own production. Solid? Very good. There are two types of inhibition that work this way. Either allosteric or non-competitive or competitive inhibition. Yes, please.
Is it also the 3-enzyme metabolic pathway?
This is just another, it's the same, it's analogous to this, but it's just meant to be a more simplified, like where the products and the intermediate molecules are all just called A, B, C, and D. It's like, yeah. It's a 3-enzyme metabolic pathway. It's what I'd say is a generalized 3-enzyme metabolic pathway. Yeah. This is a 3-enzyme metabolic pathway. So is this, but this isn't like a real one. Yeah. Cool. So check this out, right? We're going to look at competitive and non-competitive inhibition. This example right here is just showing inhibition, where D is inhibiting enzyme 1 from acting. All right, this right here is specifically isoleucine and a specific example of non-competitive inhibition. So check it out. We've got this initial substrate molecule at the top, threonine. It binds to the active site. Enzyme 1 makes an intermediate, A. Enzyme 2 makes an intermediate, B. So look, this is a 1, 2, 3, 4, 5 enzyme pathway, right? You start with this molecule, you make isoleucine. But you don't want to make too much isoleucine, and you don't want to use up all your threonine, because the threonine is going to be used in all sorts of other pathways. So far, so good? Sick. Now, in this specific type of feedback inhibition, the end product binds a site on the enzyme that is not the active site. So here's the active site right here, okay? There's the active site right there. There's the active site right there. When the active site's available, it'll just bind threonine and this pathway proceeds. In this type of feedback inhibition, the product of the reaction binds to an additional site called the allosteric site. And when it binds to the allosteric site, it changes the shape of the active site, making it incapable of carrying out this conversion here. Cool? So this is non-competitive inhibition, also known as allosteric inhibition, because the inhibitor, this end product isoleucine, is not competing for the active site. That's why it's non-competitive. Instead, it's binding to the allosteric site. That's why it's allosteric inhibition. You got to know both terms, and you got to know that they're the same for our purposes. Awesome. Awesome, awesome, awesome. So check it out. Same benefit. As isoleucine gets used up by the cell, it releases the inhibition. It allows the metabolic pathway to move forward. Cool? All right. So allosteric inhibition. There's also allosteric activation, okay, which is the same sort of process where there's this additional allosteric site, but there can be this thing called an activator that binds, and then it makes the active site become active. The reason that I put this slide up, because I'm not going to quiz you or really talk about allosteric activation, but I want to justify why do we have two separate terms, you know, allosteric inhibition and then also non-competitive inhibition? Well, because there's allosteric enzyme regulation, which is just anything that involves an additional allosteric site, an additional site, not the active site, all right? So we're talking about allosteric inhibition right now, though. There's the inhibitor, it binds there, changes the active site shape, so the active site can no longer hit substrate. Cool? All right. Now, why might you want allosteric activation? Well, maybe the activator is going to be reacted with by the product of the enzyme catalyzed reaction. There's lots of cool reasons for allosteric activation, but either way. Good. Activity to shape change. Depending on where the inhibitor binds, you can have competitive inhibition and non-competitive inhibition. Competitive inhibition, the inhibitor binds the active site. Non-competitive inhibition, the inhibitor does not bind the active site. Okay. Do you think this is going to be on your exam? I've had like a dozen slides about it, right? Yeah, exactly. It's going to be the smallest one. True or false? Yeah. Do competitive inhibitors bind the active site? True or false? Mm-hmm. Yeah. I should have a whole section of the exam that's all that. All right. So here's a generalized competitive inhibitor. It binds the active site. I'm so tired of repeating that. Do you get this? Are there any questions about this? All right, I know that was getting painful. All right, let's keep on going forward. So competitive, non-competitive inhibitors, they adjust enzyme activities differently. Yes, please.
Are inhibitors only like the end products, or are there inhibitors that are just inhibitors?
In feedback inhibition, inhibitors are the end products, but there are other types of inhibitors. Yeah, that's a great question. So there are other things that can inhibit enzyme activity, and sometimes they're things that mimic substrate molecules but can't be broken down by the enzyme that we just make in a lab, all right? And then those things just sit in the active site of the enzyme and slow the reaction from going on. Yeah, I love that. That's great. So competitive and non-competitive inhibitors affect enzyme activity in different ways, okay? So check this out. And this is going to look familiar to what you see in your lab that you're doing tomorrow, because you're going to be looking at enzyme activity, but there'll be no inhibitors involved. It'll just be you change concentration of substrate or you change concentration of enzymes. So check this out. What we've got right here, there's a constant concentration of enzyme, all right? And you just keep adding substrate at different levels. And then you measure the rate of the enzyme catalyzed reaction. If you have more substrate, then the enzyme catalyzed reaction can happen more quickly, because remember, the enzymes and the substrates are all zipping around inside of the solution in random directions. So if there's more substrate, there's more likelihood that there will be an encounter between the enzyme and the substrate. So far, so good? Awesome. Now check this out. Eventually you hit a point where you've saturated all of the active site. There's so much substrate that the enzymes are doing this. They take on a substrate and they convert it, and they release it, and there's already another substrate just right there immediately. So at a certain point, it doesn't matter that there's more substrate, because the enzymes are working as fast as they can. And you cap out, you reach a plateau in the rate of the enzyme catalyzed reaction. Okay? Now think about this, right? If you've got competitive inhibitors, okay, in the solution, if you increase substrate concentration enough, then you're going to swamp out the competitive inhibitors. Because the competitive inhibitors, they'll get in there for a second, but it's transitory, okay? So if there's enough substrate, the enzyme's more likely it keeps encountering substrate. So the maximum enzyme rate stays the same, and if the substrate concentration gets high enough, it doesn't matter that you have the competitive inhibitors there. But the non-competitive inhibitors, those actually change the maximum rate of the enzyme catalyzed reaction, because they're binding to an additional site and changing the shape of the active site. But once again, it's transitory, okay? The binding to the allosteric site's not permanent. An allosteric inhibitor doesn't hit there and then just turn the enzyme into nothing. It wouldn't work for feedback inhibition then, right? So if you've got allosteric inhibitors, then you've effectively turned off a portion of the enzymes in the solution. So it still matters what the substrate concentration is, but the plateau is going to be lower. The maximum rate of the enzyme catalyzed reaction is going to be reduced because a portion of the enzymes are not functioning. Yes, please.
And this is due to shape change?
It's all due to shape. Well, actually, no, sorry. In the allosteric inhibitors, in the non-competitive inhibitors, it's all due to shape change. In the competitive inhibitors, that's because they actually block the active site.
It blocks and it comes off and then it can block again. So that's why it
Yeah.
can reach that maximum rate, but with non-competitive, it changed the shape so that it can't keep doing that over and over again.
Points. Yep, absolutely. Yeah.
So I do have another question if we have
Yeah.
time. Could you explain again really
I know. It's
yeah.
yeah.
quick, why the main reasons why enzymes are so important, since there was a lot going on when you were saying that?
Yeah, totally. Absolutely. I'm just going to tear those speakers off. Okay? Yeah, it's like, this is the nice room, too. All right, enzymes, folks. I can't overstate how important they are. All right? You've got DNA, and DNA can encode proteins. So based on which genes are being turned on in your cells, you can change which proteins are there, right? But you can't encode or produce inherently anything that's not a protein. So for breaking down glucose in cellular respiration, for making glucose in photosynthesis, for breaking down ATP, you already talked about pumping things, but for all these metabolic reactions, that determine the non-protein components of your cells, it's all dependent on the enzymes that are present and how the enzymes are being regulated. And to a small extent, transporters that you've got, right? Because you can consume things and then transport them. But the metabolic pathways are what dictate the non-protein component of each cell type, which is incredibly important. Different cells aren't entirely determined by which proteins are there. Does that kind of make sense?
Kind of. So the enzymes are so important because they basically run everything.
Yeah. They determine what's breaking down, what's being built up, all the non-protein stuff.
And they function in the metabolic pathways, which
Yeah.
control the whole process.
The whole thing. Yeah. The big determining factor in the cells. 100%. 100%. Yeah, of course. Yes.
Is caffeine an example of an enzyme inhibitor?
Caffeine is it activates, oh my gosh, it's always is embarrassing to me because it's about, I believe what it does is it increases the rate of enzyme catalyzed cyclic AMP production, and CAMP is a signaling molecule that turns on other pathways that break down ATP and get you all jazzed up. I'm pretty sure, and I always, I'm always a little shaky on caffeine, but I am pretty sure that it does not act as an inhibitor. It instead acts as an activator. Yeah, solid. A hand, yes, hello.
Caffeine, how does that work in like ADHD? Because some people with ADHD have like go to sleep when
Totally.
they drink caffeine. So
Yeah.
I'm wondering how that
I wish I knew. I wish I knew more about that, and I just don't have a solid answer for you. Yeah. Very cool. Man, it's like that darn speaker. It is really, I got to talk to the IT folks or something, because we can't have another lecture like that. This is impossible. We have to get through the enzyme PowerPoint. We'll wrap a little early today, so just try to laser in on me for the next 4 minutes or so, okay? And we'll end a little early in recognition of the terrible noises. Here we go. Enzyme activity is so important. I've laid it out for you. It's incredibly important, all right? There are beneficial drugs that inhibit enzyme activity because runaway enzyme activity underlies a lot of disorders, okay? Also, enzyme inhibitors can kill, okay? So like sarin, okay? These inhibit this enzyme that does something to choline, and I used to remember the whole thing in the pathway, but I'm blanking on it right now. The point is that if you inhibit enzyme activity, the organism can die if you can attack a single enzyme. So one example of an enzyme inhibitor that's widely used to kill living things is right here, Roundup or glyphosate, okay? As it's actually called. So check this out. There are these here's a metabolic pathway that's really nice and simplified. You've got this precursor molecule, okay, that's going through a few steps. It branches at one point, it branches here, and it's responsible for producing some essential amino acids. And if you don't have this pathway, you don't have those amino acids. And if you don't have those amino acids, your body can't make proteins, and the organism dies, okay? So this should say glyphosate, which is the active ingredient in Roundup. And the way glyphosate works is it inhibits this enzyme at this step in the pathway and prevents the production of those amino acids, all right? So this is an enzyme inhibitor. You spray it on a broadleaf plant, and the plant dies in a few days. All right, super effective. It's really an effective product, all right? You spill some on your lawn's messed up. Now, what people have done is generate Roundup-ready plants by genetically engineering the plant to express a different version of that protein. So you take a version of that protein from a bacteria, okay, that is not inhibited by the molecule glyphosate. You engineer the gene for that protein, for that enzyme into a crop, and now that crop is a Roundup-ready crop, And when it's time to weed, you just dump glyphosate over the field and everything else dies. And you didn't have to actually put in the effort of weeding, of hand weeding or dealing with any of that. So that's how Roundup works and how Roundup Ready plants work. Roundup glyphosate just works by inhibiting a single enzyme and the plant dies. Yeah, it's pretty fascinating stuff. So what we are going to get into on Friday is compartmentalization of enzyme activity. I've already laid down for you the whole thing about, regulating enzyme activity determines cell types, and enzymatic activity is so important because it regulates these metabolic pathways and blah, blah, blah. But now what we're going to be talking about is compartmentalizing enzymatic activity into organelles. So mitochondria and chloroplasts, these two really important organelles that sort of underlie bioenergetics on the planet, all right? What do? They're just little compartments where enzyme activity is optimized. So compartmentalization of enzyme activity, it's going to be all about taking the enzymes, putting them into specific compartments, and then letting them do their thing and making the conditions ideal for those enzymes to operate. And that'll be where we start on Friday when we talk about respiration. All right, the very last slides I have for you are just, hey, put enzymes in laundry detergent to break things down. All right. Usually those are isolated from extremophilic organisms, and cofactors are non-protein enzyme helpers. Many are vitamins. All right, a lot of the vitamins that you might take as supplements, the things you have to eat that are enzyme cofactors. All right, we'll come back on this slide on Friday. So sorry, it was such a disruptive thing, those damn speakers, but we got through it. Thank you all. Thank you. A question, yeah. Yeah. Yeah, I don't know. I mean, it's like I don't think I have a way to turn it off right here. Yeah, I'm gonna end this recording real quick.